So next up is Dr. Bob Bell. Bob is a biochemist by training, having spent 23 years at Duke University doing a variety of research studies and mentoring many students, and was the founder of Duke's first biotech spin-out entitled Sphinx Pharmaceuticals. I hope I pronounced that properly. Thereafter, Bob became VP Research at Glaxo Wellcome and then spent 9 years in the venture capital arena. Along with many other ongoing advisory board appointments, Bob is also a distinguished member of the Ignite Scientific Advisory Board and will walk us through the drug discovery and development process. Bob, welcome.
I just want to say that it's really a pleasure for me to be here today and to serve on this board. And I want to congratulate Conrad and Chris for the progress that they've made to date. And I think that portends a lot of future progress. And Chris kind of jumped over some findings and that they just, flashed, they were so understated I could hardly believe it. Anyway, good work. So today I want to talk about drug discovery, development, and commercialization. And it's going to basically be a 101. And the PowerPoints that I put together were borrowed largely from pharma, and they were put in excellent shape by Nate, who helped me with them. So now these PowerPoints will describe the processes of drug discovery and development and help us build the vocabulary that's used in the industry and for communicating about these processes. And they really provide the framework for further inquiry and questions. Now, from 30,000 feet, what is drug discovery and development about? It's really about a set of scientific studies that are required to discover and develop a drug. It's really simple. It's just a set of studies that are required to discover and to develop that drug. But this complete set of studies represents one of the most challenging scientific endeavors still on the face of this planet. It's because you have to get each step, each set of studies completed in a satisfactory manner before you're going to be able to get that drug to market. And so it's a challenge, and a challenge is an understatement. It is risky. It's not for the timid or the weak at heart, because it's fraught with a difficult path. But yet, it's noble work trying to find drugs to help sick people. And it's really exhilarating when you get something that does. And for the scientists that are really drawn to these activities are, have to be labeled, and this was mentioned in your introduction this morning, Steve, is they have to be courageous. It takes courage to be involved in these activities. And they have to be committed and fight at every step to get each one of these pieces of data together. And it's really, you fight against those odds to get this medicine and to improve the human condition. Now, I think this is true for every one of you who are involved in this really extraordinary project of the Ignite project. You're involved in this fight. Now, for me, I've been involved in this fight for some 35 years, and some 23 years were at Duke, actually getting ready for the fight. And research in our laboratory there led to some interesting observations for drug discovery, including cancer research. And were those findings that led me to found Sphinx Pharmaceutical. It wasn't that I woke up one morning and said, I want to start a biotech company. I wanted to solve a problem, and the industry was not responding fast enough to solve that problem. So I decided, well, we can do it another way. You can do it yourself. And that's what caused the Sphinx to be founded. And then I really was hooked with the drug discovery challenge, so to speak. It's not that I don't like to succeed, but I'm this eternal optimist that believes he can walk a path where success can be found through all the mire and the quicksand. And so on two weeks' notice, I left Duke University's 23 years, my cushy life of a endowed chair, and a named professorship to join Glaxo to head up research. And I'm glad I did. It was a huge learning experience, and not only at the level of drug discovery, but at the level of leadership. Because now I was the one that some 600 people looked to for guidance. And this brings me to an extremely important point about drug discovery. Its complexity means that no one person has enough information, enough knowledge, enough time to actually discover and develop a drug. Drugs really are only discovered and developed through teams, and really effective teamwork is essential. And again, one of your points in your introduction I'd like to reiterate, because that is an essential hallmark of effective drug discovery. So drug discovery is not an individual sport. It's not something that the Lone Ranger off in the lab. They may generate the spark, but unless they can play in part of a team, it's not going to happen. All right, so now we can get into these, whoops, we'll go back one slide. So we're going through the process now of drug discovery. And Chris has mentioned some of this in his talk about starting with a target. And these target selection is extremely important. And integrating this into what's coming from the IGNITE program, I think is going to be a challenge, but it looks like from the preliminary results that Chris has reported that the data are going to be extrapolatable and have some immediate use, and I think that's really very important. So you need the target, and that target needs to be validated. Then you need to find the right molecule. I mean, these things sound simple and easy, but they're not. And then that molecule has to be tested in the lab and in the clinic for safety and efficacy. And the goal is very simple. It's to get it approved and to launch it and get it in the hands of physicians, available to patients, and then subject to follow-up studies. So those drugs that are successful and lead to approvals often will take 10 years, maybe longer, to get to this point. So there's a lot of work and a lot of money and a lot of data that has to be collected. So this process then actually begins with focusing on the target and understanding the disease. In-depth understanding of the disease is really essential because that's going to later on help guide the clinical trials and the clinical trial designs. And it also guides the cell biology and the in vitro tests that are going to be developed. So in this particular slide from pharma, they talk about a few years getting ready, and then you have the target, and then it's go. Put it together with some 5,000 or 10,000 compounds, start to sort through things that might be possible. And then when you get ready for the IND, It means you've taken it, you found hits, you have leads, these leads are optimized, and you come up with candidates. So the candidates are the drug-like molecules that are now to be tested in the clinic. These molecules may number 5. And then from 5 molecules that enter the clinic, 1 may actually end up being successful as a drug. Now at Glaxo, Wellcome, I went to Glaxo and was there 23 days, and then we bought Burroughs Wellcome. So I didn't know that my job was going to double overnight, but it did. So I had a chance to put together a new organization in Glaxo Wellcome and to create a new research strategy. And we wanted everyone in research to know and discovery to know what they were about. And so we came up with something that kind of sounded like a football play, 30-10-5-1. We wanted everyone to be able to say, what's your strategy? It's 30-10-5-1. That's 30 targets every year, 10 leads. So leads would be pretty effective molecules that went up against the targets, some validation of the target. And from those 10 leads, we wanted to get 5 candidates that would go into the clinic, and from those 5 candidates, we could produce 1 drug a year, 30-10-5-1. And it's pretty interesting for me now to look back at this slide, which is more current, but the number of 5 is still resident on this slide, and 1 to get that drug. So in this process then of drug discovery, it's very important to choose the right molecule. Everyone gets to play at this. Every scientist that wants to be involved in drug discovery is welcome. They can contribute at any number of levels. And then usually the search for drugs has occurred in the pharmaceutical or biotech industry. But increasingly, academic groups are becoming involved and quite sophisticated in finding drugs. And I think that's very healthy. But the net result of all of these activities in the early phases of drug discovery are to produce a candidate. And we'll talk about some of the properties of that candidate before we go into the clinical testing. So this disease knowledge can't be understated. It's extremely important to understand the disease and how it presents and manifests itself in a person, because that's going to tell you how to design those clinical trials. What are you looking to ameliorate in those patients? And I think that you because you're approaching this genetically with rare or orphan diseases, that's a great advantage, because you know exactly what you're looking at and because you're working from the phenotype back to the genetic understanding. So that gives you really a leg up. Now, Chris had mentioned that some of the targets were so-called druggable. The reason you think about druggable targets is because that's where the highest level of success has been seen historically in the industry. So druggable targets are known to all of you, like enzymes, receptors, 7 transmembrane receptors are one of the largest class where successful drugs have been discovered and developed. Nuclear receptors, those are the main catch basins. Other areas have been, kind of much less successful. If you have to block a protein-protein interaction to affect the desired outcome, those have proven to be much more difficult for a number of reasons. Okay, so this target validation is very important, and our strategy at Glaxo was to do what we called hit-based target validation. we would take from the literature, take from the thinking, understanding of the disease, a target that we thought was probable. In the past, years would be spent trying to validate it with imprecise methods. At the end, you would get through and you'd say, yeah, it's probably valid. So we decided we're going to forget all of that work. We would go for the hits directly, and we would use those hits, those drug-like molecules, as tools to validate the target in a variety of settings. And we found that to be actually quite helpful because you had a drug-like agent to use to find out whether, well, first of all, did it bind to the target? Did that change the behavior of the target molecule, an enzyme, for example? And did that have a consequence on the phenotype, the alteration in the disease process? So anyway, the end point here is to get to that candidate, and then part of building that candidate is to do a lot of preclinical testing, both in vitro and in vivo. There are lots of ways of going about this now. Animal models are improving, but yet there's still a high need for animal models that are predictive of this disorder, especially true in the areas of CNS diseases where there's a history of having models, good data in animal models, and nothing happens in human beings when the trials are done. This process is a part of the late stage of discovery, getting up to that candidate, where you also do ADME and tox and understand the bioavailability, how rapidly metabolized the molecule is going to be, what the excretion and distribution properties are. These are all properties, solubility, physical properties. All these things have to be lined up. And at this stage, you even start to think about how you would scale up the chemical process to make this molecule. Because, you know, there are certain limits on costs of goods that can go into drugs and still maintain a profit. So you want to make sure that there's going to be a large-scale process. You may not have reduced it to practice, but you will certainly have done some pilot work to make sure that you can produce that drug. And then the candidate is ready, and it goes with a package to the FDA in what's called the IND filing, Investigative New Drug Application. And following review, you can then, it can be tested in volunteers in phase 1. These are just volunteers, they're not patients. In some cases, like in cancer trials, they could be cancer patients, but most times it's just volunteers. And they're just tested for tolerability. You can do some PK work in that phase 1 and be ready to go for phase 2, where you can determine dose ranging, and then you can start to find out if you're going to get a hint of efficacy to support a very, very expensive phase 3 pivotal trial. So working with the regulatory agencies is a critical set of skills and knowledge to be able to advance these molecules. And one of the things that I like to think about and actually suggest to scientists at the very beginning of the drug discovery process is to think about what's called the target product profile. And that is basically starting to set the stage to think with the end in mind. And as we talk about commercializations, I'll then cover creation of these milestones, when to partner, and formulary challenges. So this strategic product profile is really the vision for the product and it's based on the unmet medical need in the marketplace. And this will mean that usually there will be drugs available in the marketplace that can treat certain conditions. I think you have an advantage because in most cases, as I understand, there aren't current treatments for these orphan diseases. But you need to understand what's in the market now and what the marketplace is likely to look like in 10 or 12 years when your molecule is going to be approved. And that is clearly looking through the telescope and trying to anticipate what's going to happen in those marketplaces. But the other point about a target product profile is that its purpose is to provide a format for discussions between the sponsor and the FDA that can be used throughout the development process. So from pre-IND through the NDA and post-marketing. And this target product profile embodies all of the specifics of the labeling concepts for these guidelines and to be used in discussion with the FDA. And it allows, through this dialogue, a design for the conduct and analysis of the clinical trials. And this, of course, increases the efficacy of the drug development process. So it's a tool for thinking about drug discovery and how it relates to a specific product. And I think it really helps sharpen up even the kinds of studies that are needed for that IND package. A lot of time, I think, scientists who are not used to this level of discipline are thinking about an agent, oh, it'll be good for cancer. Well, they're not thinking about which cancer, which patients it'd be good for. And it really does provide some discipline for these processes. So in terms of the value creation milestones, it turns out that every step along the way adds value. And it used to be that gene function and gene products, these were really hot commodities, and you could actually get pharmaceutical companies that would just pay for a target. Right now, pharmaceutical companies, as far as I know, are overloaded with targets and opportunities. There's a glut of them. So to get interest at this level is unlikely, except in certain disease states like Alzheimer's disease. If there were a potential target that's novel and a breakthrough could come for a huge market, you might get interest at that early level. Value is definitely added going through the hits leads candidate, and by the time you get to a candidate, you have value because you have a molecule that's ready to go into humans. And of course, most molecules will make it through phase 1, but when you get into the phase 2A, 2B trials, that's where enormous value is added because they're not only safe, but they're tolerated, and you can start to show efficacy. And that's where drug companies will actually step up and start to pay. We've seen the pharmaceutical industry shift the risk from themselves to biotechnology companies. And biotechnology companies are currently doing clinical trials that they never fathomed that they would do. And it's really putting a lot of pressure on the venture capitalists. Okay, so the rest of that is, so when you seek a partner, I suggest that you start talking to potential partners right away. There's no reason not to talk to the big pharmaceutical companies. You'll find out what they're thinking about, what their so-called franchises look like, where in 3, 4, 5 years they may be interested in. If you can start to form a relationship with them, bring them up to date every year or 18 months. And in pharma, nothing is ever the same. If you go 1 year, it'll be different. Even 6 months, it'll be different. So you have to keep checking in and talking with them. And they can give you guidance. They can say what kind of data they would need to really show interest. So that's pretty much what I wanted to say about the drug discovery process, except to say I think that the challenge of it is extraordinary. It's something that those who are courageous and brave enough to get involved and to be totally committed to, and I've seen it where people will just knock down the walls and find solutions when you're sure that the molecule's dead, it'll come back. It's that level of perseverance and commitment that's required to advance a drug and to get it developed. But it's also in that process where a scientist is able to create enormous value for patients and to leave a life of purpose and one with great satisfaction. Thank you.
Well done, a nice concise summary of something that I know is a very, very complex subject. Before we break, we have time for a few questions, and we welcome them. Rod.
Well, unfortunately, we find a lot of drugs that are withdrawn for reasons of toxicity. I know less about the ones that are withdrawn because they finally decide they don't work.
I just meant in total. How many get withdrawn for whatever reason, post-marketing?
Oh, post-marketing. Well, I don't have an exact number as a percent of the total that are approved. All we know is that as a result of these drug withdrawals, the FDA has switched into a more cautious mode where it's like safety first and putting even more demands on the drug developers. And that's slowing the process down. And of course, I'm not advocating doing things that are unsafe, but there is a time when you have enough data that the only way you're going to get it is by putting it into a larger population of humans. And yeah.
Yeah, let me add something to that. I'm Marlene Hafner, and I worked at FDA for a few years.
Yes.
The numbers of drugs that are withdrawn, and they're withdrawn generally for safety, are few. They get a lot of press, but the actual numbers are few. FDA now has something called Fast Track, where you can get your drug approved based on biomarkers of whether that drug works, and then follow them after approval to determine whether they really do work and what the safety signals are. I don't know of any drug that's been withdrawn for efficacy.
Yeah.
That's not to say that there shouldn't have been some, and that's an off-the-record comment. But it's really safety that gets drugs withdrawn. Because, and certainly with orphan drugs, you have a very small population in which a drug is tested. So if you're going to see efficacy, that drug needs to be efficacious, because no drug is 100% efficacious. And with an orphan drug, if you've got 30 people in the clinical trial, in order to show efficacy, you have to have a fair percentage that have a beneficial effect. On the other hand, you know far less about safety, again, because you have a very small number of patients that have been exposed to the product.
Yeah, thanks.
So I liked your 30, 10, 5, 1 mantra, and I was just curious, in practice, what do you think those numbers really are?
Today,
Industry.
it's probably not quite that efficient today, because those numbers were chosen for a threefold increase in productivity. And as best we could tell by our metrics, over a 6-year period after that strategy was put in place, we were on target. We were putting out the candidates. So I still think if you really get a good lead, there's a 50% chance of optimizing it if you put the right chemistry resource on it. The problems still are coming in the clinic where molecules go in and for unexpected reasons show no efficacy. And that's attributed in many cases to inadequate animal models, so that's why I made that point about the need for better models. So it's greater than those numbers, but we know that from phase 1 to phase 2, there's very little attrition in that particular phase, but it's all on the efficacy signal once you get through the safety signals. What do you think is?
I don't know. You know, there's a lot of numbers that are bandied about. 10% or fewer go from phase 1 to phase 3 to approval, but
Okay.
whether that's a real number or whether that's what we
Pulled out of a hat.
call swag, a scientific
Yeah.
measure, what best. I don't know.
But the reality of it is, with all of the biotechnology, all of the advanced instrumentations, highly sophisticated ways of looking and living cells, improvements in chemistry and making all kinds of different molecules, improvement in animal models and cell-based models. The discovery process is still not very efficient process. And it's real tricky. We used to say there were drug finders, and these were people that had a nose for it. And they were champions, and they could keep their projects along. And of course, by definition, if your project's alive, it has a chance of going all the way versus all those that are killed. But I think the consolidation in the industry has also really, really hurt the drug discovery efforts. In spite of saying that you can do things on a larger scale, this and that, and there's a huge amount of disruption in that pre-phase, and the knowledge, the tacit knowledge base that's lost, that just walks out the door. And the disease base.
Paulin?
Yes, I have a question, general questions. There has been a school of thoughts been talking about this, the era of dying blockbuster drugs. And how true is that in reality?
The era of?
No more blockbuster drugs would be
No
developed.
more proprietary drugs?
Blockbuster.
Blockbuster.
Drugs.
Well, I don't think that's it. Blockbusters will be there. The industry will try to make as many as they can. In fact, blockbuster drugs emerged. The guys in the marketing division and the commercial side would try to look at a market. For example, when like Zantac came out, the predecessor was a SmithKline drug. And there, they thought maybe it'd do $40 million a year. And then later they looked at it again, and maybe it'll do $100 million a year. Well, it did over $200 million the first year and went up beyond that. So for indications that are known in the marketplace, you can pretty much predict the market. But new entries into the market will often drive the market. The market is a lot larger than people can sit around and read tea leaves and figure out. So with your question about blockbusters, I think, of course, that blockbusters will exist, largely because they're a consequence of providing effective therapies to people around the world, and the sales will just drive it up. The idea of starting to drug discovery only to find blockbusters is fraught with a lot of difficulties. But on the other hand, you still want to understand the disease enough and, you know, how many patients there are so that if you make a drug that's quite effective, you can make a serious amount of money. It used to be that the first drug to market made all the money, but that hasn't been true for years now. Often the second and third, it's the best molecule in the market that makes the money. So there are a lot of implications for that, but I don't know anyone saying they're only practicing a blockbuster strategy. Look at the interest now in orphan diseases. That is completely on the other end, but yet there's a considerable interest in pharma. And largely, I think, because pharma thinks they can get a lot faster approval and get to revenue quicker. Yeah.
Okay, thank you very much, Bob.
Sure.
We're